Saint on, and it's great that the first speaker is already standing there. But it's a great pleasure to announce Andrea Geisz, who actually used to be a student and a postdoc in my laboratory, and then she became independent at Boston University, where for the last 3 years she has been working on a brand new project which she will present today about the role of cathepsin B in pancreatitis. Andrea, floor is yours.
Thank you, Miklos, for the kind introduction, and I'm also very thankful for you and for Peter that I got invited to this amazing meeting and I can present my work here. And as you mentioned, I'm going to talk about a project, a very recent project of mine, which is intended to investigate the role of cathepsin B in pancreatitis initiation. So
the microphone on, because it's very.
It might be too low.
I'm short, so is it on? No, it's better.
I hold it up.
Okay.
I hold
It's
it
not
up.
on.
It's not on. Cannot be on.
I think no.
This has to be me. Yes, yes, thank you. Okay, so the initiating pathogenic events in pancreatitis have been extensively studied, and premature activation of digestive proteases, trypsin in particular, emerged the most plausible mechanism for disease onset. And in early studies using rodent models, in experimental studies where they induced pancreatitis with a cerulean, they found that lysosomal enzymes like cathepsin B actually gets missorted during initiation of pancreatitis, and they get, or it gets redistributed into a zymogen-enriched granule along with trypsinogen, which would set the stage for pancreatitis initiation. And then this was backed up with a study which investigated a cathepsin B knockout mouse model. And once they induced pancreatitis in this model, they found that trypsinogen activation was actually diminished and pancreatitis severity was mitigated. And the third study I'm showing here was actually another proof that when they inhibited cathepsin B pharmacologically, they found again reduced trypsin activation and ameliorated pancreatitis responses. And what I'm not showing here, that there was a study which identified cathepsin B mutations or mutations in the cathepsin B gene in association with chronic pancreatitis. And this was done in the Indian population. However, this could not be replicated by European cohorts. So another group of people would say that trypsin gets activated through a trypsin-mediated mechanism, trypsin-mediated trypsinogen activation, which we call autoactivation. And actually, there is multiple proof for this mechanism. So the first one was a very defining paper of the field where the Dr. Whitcomb identified the disease-causing mutation in hereditary pancreatitis, which was the R122H mutation. And then there were a bunch of subsequent human genetic studies identifying more and more mutations in the trypsin or in the trypsin degrading chymotrypsin C or the trypsin inhibiting SPINK1 gene. And then when we looked at what's happening in a test tube, we found that a common phenotypic change for these mutations is an increased propensity for trypsin autoactivation. And then very recently, we actually proved this in vivo as well with this preclinical mouse model where we induced trypsin mutation in the activation peptide of the trypsinogen, which caused a robust increase in trypsinogen autoactivation. This was actually 50-fold. And then these guys developed spontaneous acute pancreatitis and progressed very quickly into chronic pancreatitis. So while, to summarize, why the important role of trypsin autoactivation was proved by not only human genetic studies, biochemical studies, but also animal models, the cathepsin B-mediated trypsin activation has very little proof, and mainly the main evidence is coming from animal models, actually. So we wanted to address this knowledge gap. We wanted to find out what's the role of cathepsin B, and we wanted to approach it from two angles. So in the first part of my talk, I'm going to be showing some data on new mouse models where we actually made a mutation in the trypsinogen activation peptide, where we wanted to prevent trypsinogen autoactivation, but we wanted to make this mouse trypsinogen very sensitive for cathepsin B-mediated activation. And then in the second part of my talk, I will be showing some data on what happens when we take out cathepsin B as a component and we knock out the cathepsin B gene, how this would affect pancreatitis responses. So this was tricky before to achieve, to separate these two mechanisms, because the trypsinogen activation peptide is actually cleaved off at the same cleavage site by enterokinase under physiological conditions in the duodenum, but also by trypsin and by cathepsin B under pathological conditions within the pancreas. So what we did, and the study was done by Alexander Demchak, and what we did, we engineered mouse trypsinogens in a way that we eliminated the trypsin cleavage site by mutating this into glycine. And then we did like an aniline scanning in the trypsinogen activation peptide to find mutations which could boost a little bit more the cathepsin B-mediated activation. So once we were done with the mutagenesis, Sandra purified these enzymes, and then she did a bunch of biochemical assays. And what she found that there was a mutation called D23A K24G, which caused normal activation by cathepsin B over the pH range of 4 to 5. And there was a mutation which caused like a 15 to 18-fold increase in cathepsin B-mediated activation. And importantly, all these or both of these mutants were defective in autoactivation or activation by enterokinase or any other proteases. And so then these two candidates proved to be good models for mouse generation. And then what we did was using classic homologous recombination-based techniques to induce or introduce the mutation in the exon 2 of the mouse trypsinogen. And then since the knock-in allele contained like a scar sequence of one of the LOX3 sites, I'm not going into details, but when we were done with model generations, we wanted to check how this would affect the expression of the T7 trypsinogen of the mouse. And I probably failed to mention that that's the cationic trypsinogen in the mouse. And so what we found with RT-PCR that there was no change, so T7 expressed normally, and we did also protein assembly analysis, and we found similar expression for both mutants and for the Black 6 controls. So this was a good thing. So then we went ahead and we wanted to induce pancreatitis, and not only induce pancreatitis, we wanted to measure intrapancreatic trypsin activities. And the way we do that is that we induce or we inject the animals with cerulin one time, and 30 minutes after the cerulin injection, we sacrifice the animals and we measure trypsin activities with fluorogenic substrate. And it's very important to sacrifice the animals within 30 minutes, because at this point the pancreas is still intact and there's no necrosis present, and there's no infiltrating inflammatory cells, which could be a confounding factor. And with acute pancreatitis induction, we did a 10 injection model hourly injection, and then we sacrificed the animals an hour after the last injection. And we just measured very classic, basic pancreatitis parameters like edema by measuring pancreas weight. We measured plasmaamylase and looked at histology. And so when we looked at intrapancreatic trypsin activation, what we found was very encouraging, that while in the D23A, K24G, we saw an increased activation in both strains, actually, when we looked at Black 6 or the mutant one. And this was very similar to each other. And this was already good because this way we could see that the cathepsin B alone can induce significant trypsin activation. But when we even boosted further with this mutation, in case of the D22A, K24G, we saw a threefold increase in intrapancreatic trypsin gene activation. So we were very excited that we were actually hoping that this could lead to spontaneous pancreatitis phenotype or increased propensity for cerulean-induced pancreatitis. And this was not actually the case. So we had to induce the animals with cerulean. And on the upper side of the slide, you can see data from the normally activating mutant. We measured pancreas weight. These are actually the same. It's just in the middle, you see the normalized values. So in the first case, pancreas weight was very similar to the Black 6. Plasmaamylase levels were very similar as well. But in the second case, we got the same. So basically, there was no change in pancreatitis severity, which was unexpected. So when we looked at the histology, we see no change again. That hematoxin and alzine stains pancreatic sections look essentially the same in case of both mutants compared to the wild type. So as a summary of the first part, I'd like to say then that trypsin activities were significantly but similarly increased. Significantly increased upon cerulean injection in a normally activating variant, which was very similar to the Black 6, but this was even more pronounced in the D22A, K24G mutant. However, this did not translate into more severe acute pancreatitis phenotype. So then we looked at what happens if we knock out cathepsin B from the mouse, whether this would affect pancreatitis severity. And if you remember at the beginning of my talk, I mentioned that when they did that previously, they found actually that, yes, trypsin activation was decreased, and they saw decreasing levels of lipase amylase, and necrosis was also decreased about 50% in the cathepsin B knockout animals. So then we generated a new cathepsin B knockout mouse because we found this. This was important because the one which is commercially available, it's on a mixed genetic background, and we all know work with animals and trying to induce pancreatitis in animals that different mouse strains have different susceptibility for pancreatitis development. So we felt this was important to generate a cathepsin B knockout mouse on a pure Black 6 genetic background. And we did that with the help of CRISPR-Cas9 technique. And basically we deleted like a 12,000 nucleotide sequence from the exon to the exon 10 of the mouse cathepsin B gene. And then we wanted to make sure that we did the right thing. So we checked mRNA expression. And in homozygous animals, there were essentially no mRNA expression detected by RT-PCR. And not surprisingly, we did not detect any cathepsin B protein as well. So then again, we went back and checked whether what was happening in case of intrapancreatic trypsin activation. And essentially, this was decrease in homozygous cathepsin B knockout animals. So we were again excited that, okay, what's happening once we induce pancreatitis? And actually, what we found was contradictory to previous data, because we found that pancreas weight was very similar to the Black 6. This was even a little bit elevated once we measured just milligram values, but this difference disappeared when we normalized to the body weight. Plasmaamylase was also very similar. And then when we looked at the histology, we essentially saw no change compared to the Black 6. mouse. So as a summary, I'd like to say that intrapancreatic trypsin activation was significantly reduced in the cathepsin knockout animals, and the severity of sertraline-induced pancreatitis was very similar to the black six. And this brings me to a conclusion, actually, that cathepsin B induces significant trypsin activation, even when autoactivation is not there. However, cathepsin B mediated trypsin activation does not determine disease severity. And thank you very much for the attention. I'd like to acknowledge Miklós, my mentor, for all the guidance, and Alexandra Demchak. She helped with the biochemical studies, identifying which mutation we should generate in the mouse, and then my previous postdoc, Anna Orokova, and my current one, Tantra, who are helping a lot with executing the animal experiments. Thank you very much.
Question?
Congratulations to this excellent talk. Actually, I wonder because when we did this trypsin experiments, and you were involved also in the trypsin one, and we also looked at the BRSS1 mutation ones. You already know my question, that in different pH levels, how they affect it. Do you have any data about or any ideas? Because even from the colocalization, the pH starting changing inside the granulomes, inside the ductal system, inside everyone. So how this process is pH dependent?
So these mutants would not get activated by trypsin on any pH. We just tried 8, but pH 8, but we blocked basically the trypsin.
And if it's acidosis?
So for cathepsin B, we of course we check the pH from 4 to 5, and then there were little differences, but yes.
But there were no significant.
I mean, probably the 4 change went to 18 to 15 in case of the increasingly activated mutant, but the other one, the first one, the boring mutant, that had no change in it.
I was going a little bit. I mean, just a simple question for a simple-minded person who is not a biochemist. I mean, if you mix cathepsin and trypsin and do this in the test tube at a range of pH values, is that simple experiment done? And what happens in those cases?
But so again, these would not, if I understand well your question, so we are completely blocking the trypsin cleavage side because we introduced a glycine instead of lysine. So there's no activation by trypsin whatsoever, or by chymotrypsin or enterokinase.
Yeah, but I mean, in the simple case where you simply have trypsin and cathepsin and haven't done anything else, I mean, then cathepsin would activate trypsin or not?
Oh, okay. That's a very good question. I don't think it would.
It's asking if trypsinogen is activated by cathepsin B.
Yes, it would.
Thank you.
But let's say in a very simple test tube experiment, and then systematically investigating the range of pH values. I mean, there is a pH value at which it would not work at all, and there would be some kind of maximum.
Yes. So I think above 5, it would not work. I haven't tried personally, but we have this. Yeah, we always set these experiments from 4 to 5 because that's.
Cathepsin B needs acidic pH.
Yeah.
So if you go towards 6, 7, then you lose activity rapidly. So it works but Andy showed 4, 5 in that range. Maybe 5.5, you get some activity. And Julia did many of these assays too. So you have to be at acidic range for that.
Can I ask a question? I'm
Yes.
on the Zoom. Did you guys try to cross the mice to try to knock out both the trypsin and the cathepsin and to see if the mice will then be really protected?
So, yeah, so for instance, we haven't combined yet these mutations, but yes, we combined the D23A mutation, which would cause a 50-fold increase in trypsinogen autoactivation, crossing with the cathepsin B knockout, and we were wondering that this would at least delay the disease onset, and nothing is happening. The mouse gets sick as early as 3 to 4 weeks of age. So it really seems like cathepsin B has no impact on whatsoever, no protective effect.
Thanks.